Today I'm going to talk about measurements of human brain structure conducted with MRI and how we're using some pretty simple principles from statistical learning to understand these measurements, interpret these measurements. This is all done in the context of an open source software project called dipy. That stands for Diffusion Imaging in Python. I'll explain what diffusion imaging is in just a second. First of all, just to mention, you can see these slides over here. You can follow along if you want, or review these slides afterwards. Just to give a little bit of context, I'm part of an institute at the University of Washington called the eScience Institute. We're a group of researchers from a variety of fields. This institute is very interdisciplinary. And together we're trying to tackle data-intensive research across a variety of fields. And that includes integrating together understanding from fields like statistical learning into neuroscience and the application of these ideas in software that other scientists can also use. So this project falls pretty squarely under these ideas, but is also the continuation of research that I did as a postdoc at Stanford University in Brian Wandell's group there in the Department of Psychology. So I should give credit there as well, and I'll mention my collaborators at the end. So the research that we do is based on an understanding that's actually pretty old. These gentlemen in the center of this image are all neurologists, and that in the 19th century started noticing patterns that recurred in patients where damage to connections between different parts of the brain were associated with very particular kinds of behavioral deficits. So connections between, say, the temporal lobe over here on this side of the brain. So this is a human brain. This is the front of the brain. This is the back of the brain. We call this part here the temporal lobe. And one of these findings was a very important finding that a connection between the temporal lobe and what we call the frontal lobe over here in the front leads to particular damage, for example, in the ability to speak. Okay, and their insight from that was that our normal behavior is supported by the existence of brain connections. They understood then that the brain is a big network that is connected through connections between cells that exist on the surface of our brain called the cortex through long-range projections that connect those different parts. This was even before people knew exactly what are the substrates, what are the nerve cells that connect between them. But they could already at that point know that normal behavior is supported by connectivity. Now this is, again, a human brain. In this case, parts of the temporal lobe and parts of the back of the brain, the occipital lobe, have been dissected out. This is a view from the bottom of a brain. So we're looking, again, this is the front of the brain. This is the back of the brain. And we're looking at the brain from the bottom up. Here, this part has been dissected away. And what you can see is that the white matter is organized in very large bundles. Now, each one of these bundles is not a single cell that is connected. It's rather thousands, many thousands of cells that are connected through what we call white matter fascicles. So we call this white matter because the internal part of the brain contains a lot of fat and is brighter than the outer part of the brain that we refer to as gray matter. And so these bundles are the bundles that support this normal functioning of the brain. But we also understand now that they're not just passive cables. These brain connections change with development. They explain individual differences in behavior. So if there are differences in the size of these cables and the extent to which they change with development, that can have an effect on behavior. They adapt with learning, so they can change even within a particular individual over a long time in development, but also quickly with learning of new information. And this has, of course, clinical significance because damage to these parts of the brain has an effect, as I mentioned before, on our behavior. The only way that we currently have to probe these connections in a non-invasive manner in humans is magnetic resonance imaging. Magnetic resonance imaging allows us to measure many different kinds of aspects of brain function. We can measure neural activity with a method called functional MRI or fMRI. We can measure the anatomy of the brain, the gross anatomy of the brain, through methods of structural MRI. And there are many other measurements that can be done with MRI. And specifically today, I will be talking about a measurement called diffusion MRI, which allows us to probe the brain connections in vivo, in a living human brain at millimeter resolution. Diffusion MRI relies on the fact that water exists within all these different parts of the brain. And if we zoom in on one of these things, so now we're zooming in a microscope, this is a microscopic image, on one of these bundles, they're made out of a lot of these tubes. And each one of these tubes really is a single cell. The part of the cell that connects different parts of the brain, the part of the cell that projects forward into other parts is called the axon. And these are axons of individual cells wrapped together in one of these big fascicles. And there's water everywhere in those parts. Now, here's sort of a toy example of what this might look like. If we had a cell that's completely round, water would be free to diffuse within the cell in all directions equally. And we'd call that isotropic diffusion. But if you think about one of these tubes, water is now free to diffuse along the length of this tube, along the length of the axon, more than across the boundaries of the axon. And this kind of diffusion is anisotropic. And we can measure this anisotropy. So here it is in the tubes themselves, right? There's more diffusion along the length than across the boundaries. And the way that we can measure this anisotropy is by the application of magnetic field gradients across the brain as we're measuring making the MRI measurement. That's the main principle of diffusion MRI. So here in this panel, what you see is the rotation of the gradients around. This here is a slice through the brain in an MRI image. It's the entire brain, so here's the front of the brain, here's the back of the brain. This is called the horizontal slice through the brain. This here is a sagittal slice, so it's a slice straight through here. What we're looking here is the middle of the brain, so here again is the front, here's the back, here's the top of the brain and the bottom of the brain. This here is an axial slice, so it's a slice through like this. And this slice, this is the left side and the right side of the brain, the top and the bottom. All of these slices are through the middle of the brain. What you can see is that as we rotate the gradients around, the measurement is sensitive to diffusion in different directions. So it becomes brighter when diffusion is restricted in the direction along which the gradient is oriented. So if you see when this is oriented like this, it'll come back again. You'll see that it becomes bright in this region here. And maybe when the gradients are oriented like that, it'll be restricted around these parts of the brain. And this restriction indicates to us, or this brightening of the images indicates to us in which directions there is restriction of diffusion of water. And that indicates to us where diffusion is anisotropic, but also in which direction this anisotropy exists. Let's look at this in a little bit more detail. So here is a measurement from a single location in the brain. We divide up the brain into these boxes of roughly 1 millimeter on each side, and those are called voxel or volumetric pixels. This is a measurement from a single voxel, and each one of these spheres represents a single gradient direction. So what you can see here is that, the distance from the center of this cloud is the distance, the estimated distance of diffusion along that direction. So when the gradient was applied in this direction, for example, that you see right here, water diffused pretty far, but if it was in this direction, water diffused pretty closely. So in this case, diffusion is anisotropic, and it's anisotropic along a particular axis of diffusion. Now we can take and build a simple model, and that's the basic operation we first do with this kind of data, is to build a simple model of this diffusion. One of the classic models that's been used in this field is called the diffusion tensor model. The diffusion tensor model approximates this diffusion as a three-dimensional Gaussian distribution. So there's how much variance is there along each one of the axes of space and how much covariance is there between the different axes. And when you build such a model, you can say here's the smooth approximation of this data by the model. You can see here this is sort of an ellipsoidal shape. So there's more diffusion along this axis than, say, along this axis. So this is the first step in analyzing the data, is modeling diffusion in every single location in which the measurement was conducted. This is very useful because we can estimate certain physical properties of the tissue from this. For example, here is, again, a horizontal slice through roughly the middle of the brain. You'll see that there is, we've been able to extract out of the model an estimate of mean diffusivity. So wherever there's very low diffusivity, water can only move very close to the center. That means that the tissue might be much denser there. You'll see that there are these areas of very high diffusivity. These are actually holes inside the brain, cavities inside the brain called ventricles that contain only water. So tissue is very low density there. It's almost purely water, and diffusivity is very, very high in those regions. Another kind of statistic we can derive from this kind of model is the fractional anisotropy. So this refers to how pointy this ellipsoid shape is, right? So if it's very, very pointy, we say that anisotropy is very, very high. And you can see that anisotropy, again, along in different regions of the brain, anisotropy varies quite a bit. Here, where we are in the core of the white matter, where there really is a dense pack of these white matter fibers, anisotropy is very, very high. And that's because water is really restricted in one direction, or rather free to diffuse along really one direction, the direction of the fibers in those locations. You can see that this forms an interesting pattern in the brain. Finally, we can derive from this kind of model, this diffusion tensor model, we can derive the principal diffusion direction. Again, this corresponds roughly to the axis of this of the highest diffusion, the highest variance of diffusion. And we can use these directions in order to now estimate connectivity. So that's the next step that I will show, is how we estimate from the diffusion to tracks that connect different parts of the brain. Again, I show you a horizontal slice through the brain, and I'm going to zoom in on this little box. When we zoom on this box, now you can see the voxels, the edges of the voxels, these little 1 by 1 by 1, or in this case 2 by 2 by 2 millimeter boxes inside the brain. We estimate one of these diffusion tensor ellipsoids in every one of these boxes. And now we can treat the principal diffusion direction of every one of these boxes as a vector and propagate a streamline through this vector field, like this, to say that there might be a connection that goes through this part of the brain in this manner. It's just connecting the dots from one voxel to the next. And when we do this many, many times here through this frontal portion of a single individual's brain, we might get an estimate of sort of larger scale anatomical structure, anatomical connectivity. In this case, these fibers all go through this yellow region of interest that we call the corpus callosum. This is a big bundle of fibers that connects the two hemispheres, the two sides of the brain. So this gives us a model of the anatomical connectivity across the hemispheres in this part of the brain. Now, all the steps that I've shown you so far, we do using dipy, and I'll just sort of talk a little bit about dipy, and then I'll present the problems that we try to solve using statistical learning and talk about that a little bit. DIPy is part of a larger community that we call NiPy. NiPy stands for Neuroimaging in Python. It's a very widespread community worldwide of people who are practitioners doing research in neuroimaging using Python. And using a variety of different kinds of measurements and different kinds of software, a lot of projects around this community. DIPy itself, diffusion imaging in Python, was started by Eleftherios Garfalidis, who's now a postdoc in Canada. There are contributors from at least 6 different countries and many, many different labs. This graph shows the growth in contributions in this project. It's kind of a nice graph to look at. There was some kind of an explosion in 2013 and been growing steadily ever since. The library contains implementations of many algorithms used both for this tracking that I just showed you and for reconstruction, the modeling of individual voxels. It also contains other modules for other kinds of computational neuroanatomy tasks. So we're using DiPy to do these things. This is all looking pretty great so far. You might ask yourself, so what is the problem? Well, Diffusion MRI has a big challenge in validating its results. For example, if you use two different algorithms, you might get quite different views of the brain. These are two different algorithms that both apply to the same data. And when you use those different algorithms, you can get either a very narrow view. So this is the same connection that I talked about at the beginning between the temporal lobe and the frontal lobe. It's called the arcuate fasciculus. It plays an important role in producing speech, in understanding speech. And depending on the algorithm that you use in this particular individual's brain, you might think that the arcuate fasciculus is this narrow, small bit that's fairly focused in its ends, or that it's this sprawling big thing. And the pattern of connectivity at the level of the cortex is also quite different. It's either very focused and narrow, or really connecting very large swaths of cortex. So how do we know which of these models is a better model of the brain? To answer that, we've taken a statistical learning approach, something we also call in vivo validation. In vivo in contrast to ex vivo. Ex vivo would be the anatomical dissection that I showed you before. Some have said you can take this brain and you can open it up, right? You can dissect this brain and look and see which one of these solutions is better. But we don't actually want to do that because A, it would be very hard to find volunteers willing to go through that, and B, the dissections themselves and anatomical methods also have their own confounds. They're not representing necessarily a ground truth, they're just representing another kind of method of measurement that has its own errors. Instead, we want to validate our models against the measurement itself. And to do that, we use a cross-validation approach. In cross-validation, we make one measurement. So here is, again, 150 different directions of gradients. It's the same as the measurements that I showed you before as a cloud. Now I just show them as a series. We can make a second measurement in the same location in the brain, in the same individual, and we can compare these measurements to each other. This gives us the test-retest reliability of the measurement itself. For comparison, we can fit a model to one of these signals, and then try to predict from the model the second measurement. And that's called cross-validation. This allows us to compare model performance in cross-validation to test-retest reliability. We can distill down the differences into one number that we call the relative root mean squared error. This is the root mean squared error of model prediction in cross-validation relative to the root mean squared error of test-retest reliability. And if the relative root mean squared error is smaller than 1, that means that model predictions are better than the SNR of the measurement itself, and we consider that a good model. If we manage to reduce the noise further than making another measurement, we consider modeling a worthwhile exercise relative to, for example, measuring another measurement. In DiPy, we've implemented a very simple cross-validation API. It is the user enters a gradient table that tells us which gradients were measured, which directions of diffusion were probed. And then some reconstruction model is generated, we call that using this measurement. So at this point, there's no data yet. Once we input into this model, we use the model fit method on this data, that gives us a fit object, a reconstruction fit object. And this reconstruction fit object then has a prediction method that can be applied again to either the measurement gradient table, the measurement scheme that we used initially, or to another measurement, extrapolating out of this measurement to another measurement of some unseen data. For those of you who've used scikit-learn, this will look pretty familiar. It's a very similar API, but notice that it embodies knowledge about gradient tables, about these particular data. So it embodies the scientific knowledge as well as an API of sort of general statistical learning. For example, for the tensor model that I showed you before, the simple ellipsoidal approximation of the data, this is what the code would look like. user would instantiate a model using the GTI tensor model object. Then the model would be fit to one data. A prediction would be generated again on the gradient table. And then this prediction would be compared to a second measurement for cross-validation. We can then compute the relative root mean squared error by comparing the model root mean squared error to data reliability. Again, test-retest reliability. And that gives us the relative root mean squared error. And this is what it looks like for the tensor model in a single individual. You can see that the relative root mean squared error is green in a lot of the brain. So actually, in most kinds of measurements that we make, we find that the tensor model is better than test-retest reliability in about 97% of the brain. I've actually focused here on the one part of the brain where test-retest reliability is actually worse in the tensor model, where the tensor model errors are larger than test-retest reliability. And we actually know why the tensor model doesn't perform very well here. That's because this part of the brain contains the crossings of several different large bundles of fibers. The corpus callosum that I showed you before that crosses between the hemispheres, but also the corticospinal tract that connects the cortex with our spine and allows us to control muscular motion, and other fibers that pass through going front to back. And that's the reason that DTI, or diffusion tensor imaging, this tensor model, doesn't perform very well there. It can't really handle crossings of fibers. But other models that do take into account this notion of crossings of fibers can actually perform quite well there. And we have models that account for the data are better than test-retest reliability in 99.9% of the voxels in the brain. So we actually have quite good models for this kind of data. Now, you might ask, what if you've only measured once? Most practitioners only measure once. Well, we've developed a k-fold cross-validation API that allows people to provide us with their model, provide us with their data, and then what kind of split they want to do on the data. And they can then use this API. Part of the magic that makes this API able to work across different models is the fact that we've implemented similar fit and prediction API across the different models. Now, this notion that we can compare the model that we have of the brain to the data itself can generalize also to a broader, larger scale. So as I showed you before, different algorithms produce different images, not only of the signal at the local scale, but also of connectivity at a larger scale. To be able to evaluate these fascicle models, Franco Pistilli and others in Brian Wendell's lab when I was a postdoc there, we developed this method called linear fascicle evaluation. The idea is that you build a forward model from the tracks to the measured signal. Here's the idea. Let's say that we've estimated one streamline over here, and then another streamline over here, and we want to validate these streamlines against the data. We can take these streamlines and create a forward model that tells us what the predicted signal would be from this fiber by itself. And then another column is in this prediction would correspond to the predictions of this other fascicle, this other track. As you can see, in some places there's no prediction at all because those are parts of the brain where this streamline doesn't pass through. So there are some places in which there are predictions from both fascicles and parts of the brain in which there are only predictions from one of the fascicles. We can then do this for many, many fascicles. We get millions of fascicles at a time for these estimation algorithms and compare it to the data as it was measured in the scanner. This forms a large linear model. Here, X is the matrix of all the predictions from all the streamlines. Beta is a set of estimated weights that we will estimate through a procedure of model fitting. And Y is the data that was actually measured in all these locations in the brain. And we solve that for beta larger than 0, so for non-negative weights on each one of these fibers. Now, part of the challenge of doing this is that the shape of this matrix ends up being rather large. As I said, there are millions of these fibers. They go through millimeter scale measurements across entire brains. It becomes a pretty large problem. Here's what the code then looks like in DiPy. It has a very similar structure to the structure that I showed you before for the models of individual voxels. So we create a fiber model. So this is the life module, linear fascicle evaluation module. We tell it about the measurement that we did, and then we fit this measurement using both the data that was actually measured and the tracks that were estimated from this data using one of these tracking algorithms. And then we can, again, so in the fitting procedure here, what happens is the fitting of this large linear model, the estimation of these non-negative weights that I described before. Once the prediction is done, or once the fitting is done, we can actually go and try to predict what would the signal look like and confront that with the data itself, estimating the error of these tracks relative to the signal. But this also gives us another kind of aspect on this, which is that it allows us to select which of the tracks actually fits the data. Those will be the tracks that received a weight larger than 0. So in practice, what happens is that many of these weights go to 0, and we can reduce the amount of data, reduce the amount of tracks that we get a lot by looking only at the tracks that have weights larger than 0. To summarize, I've told you today how we measure brain connectivity using diffusion MRI. Told you a little bit about the dipy project that we're using in order to analyze these data. In particular, I've focused on the validation problem and this notion of in vivo validation, how we're using statistical learning in order to validate the models that we estimate for connectivity of different parts of the brain. I'd like to just mention my collaborators on this work. On the dipy side, Eleftherios, who started the project, Stefan van der Walt, who is now being mentioned for the second time today, if you noticed in Josh Bloom's slides earlier, who was involved in this work, and Bago Amirbekian. There were many other collaborators on dipy, but I'm mentioning these people in particular who were involved in this statistical validation effort. And then at Stanford, during my postdoc, I worked in Brian Wendell's lab. Franco Pastiglia was the lead author on this linear fascicle evaluation approach. And then these are all my contact details. If you'd like to talk about this, you can get in touch with me, and I'd be happy to take questions about all this.